Pedro was so kind to be flexible and step in today. Thank you for that, Pedro.
Okay, thank you very much, and thank you for the invitation to be here. It's a pleasure to be always in the Catch Welfare platform. I know I'm between you and the coffee, so I'll try to go as fast as I can through my slides. So if you don't understand anything, you can then ask me in the coffee break. So I'm presenting the Carefish Catch project, which just ended, and I'm just showing you part of it, which relates to how we went about to measure welfare, mostly in the process of stunnings and slaughtering while on board. And, but there's other aspects of the project that you can see in the posters outside, and that I'll mention while we go through the presentation. Okay, so of course, being a project, this is a consortium of several groups, and these were the people that were more involved in this part of the group, but we could not have done this without the other ones. And we focused mostly on artisanal fisheries, so small-scale fisheries. But just to give you an idea, fisheries now account, and probably this is a summary of many things that you already know, but we're catching, well, this amount in million tons, and this actually reflects in about 1 to 2 billion individual fish. And if we're talking about welfare, of course, we're talking about the individual. The other point I want to make here is that a lot of these fisheries, so 40 to 60%, depending on how you use the fish, comes from small-scale fisheries. And 90% of the people involved in fisheries are involved in small-scale fisheries. So it seems small-scale, but in fact it's not that small. And we can talk a lot of things about welfare, but for many of these people that are involved in small-scale fisheries, small changes are actually big changes for their daily practices. And so that's also where we have to be acting. We have to be changes that are feasible and that they can accommodate in their daily practices. So the idea behind the project was actually to promote several dimensions. Of course, the ethical dimension on the account of welfare of animals, but also of of the fair trade of these small-scale fisheries. A conservation dimension, we've been talking about bycatch and survival of bycatch, and of course there's a dimension there on the resources and the biodiversity and the habitats. Food quality and safety, of course, again, welfare promotes better food quality, but also scientific knowledge, and as we've seen today in these presentations, technology knowledge. There's a lot to be done, and there's many opportunities here to understand how fish work, but also to understand how we can improve the gear. So, in fact, Carefish's main goal was to promote better fishing standards in the small-scale fisheries. And I also have to say that Bill Heinz-Peter, yes, I'm smelling it correctly, it was also a great part of the origin of this project because it's involved in small-scale fisheries. Okay, so to go a bit faster, so these were the groups involved in the consortium, the partners in the consortium, and you can see it was actually quite a overarching project because we wanted to, in the end, provide operational indicators, welfare guidelines, certification criteria, technical reports and publications, of course, and for that we had the expertise of different partners. And again, many of these goals are reflected in the posters outside that you can talk to Faust and Vignesh about them. Of course, this is not new. We know, though, that fish feel pain, they get stressed, etc. They have a lot of these behaviors that we can measure. The question is, how do we measure them? And we try to use these welfare assessment approaches, where you can, of course, look at the biological function and the emotional, in brackets here, approach. Of course, when you're talking about fisheries, it's difficult to look at the natural living approach because they will not, of course, perform as they naturally would do. But the challenge is how to then apply all of these criteria to fisheries. And so we've been talking also in this session already about how can we quantify welfare loss, what are the critical steps in welfare loss, and of course we know that not all fishing methods have the same impact, not all species will suffer the same. So it's a big challenge to identify the procedures that we can improve, that we should eliminate, and the ones that we should promote without having realistic or without having scientific evidence-based indicators. And of course, you will talk about the strategy about this tomorrow by Mike. But so what we need to know is not only what we could measure on board, but what was going on in the water, and that's the gap that we still have to do. So because soaking times might be different for different fish, different species or different sizes may have a different relationship with the gear. We don't even know what happens to the other fish that are in the proximities of the gear. The ones that are being scanned, in the sonars. What we actually do, or we were able to do at this point, was to measure what's going on when they arrive on board on these small-scale fisheries boats. And again, as I said, this is part of the risk assessment framework that's going to be presented tomorrow. So we had a few case studies, bottom set long lines, gill nets, long lines, and purseines. This was done in the southern coast of Portugal with local fishermen, and you'll see it's really small scale in some cases. We used vitality and scale loss assessments, observation, and behavioral assessments. So we made a simplified scale that we could measure on board, from non-responsive to high responsive, which we call level 1 and level 4. And of course, level 1 is non-responsive. We don't know if they are dead. And also our statistical models, they don't like 0, so we kept the 1 in there. Scale loss, also different discrete levels. And we use physiological indicators, cortisol, glucose, lactate, and osmolality, because they are relatively easy to measure, and they give us different indications. So cortisol for stress levels, intensity and duration of stressors, energy mobilization. So to meet metabolic demands, we use glucose. Exhaustion and anaerobic respiration, we use lactate. And hydromineral balance, we used osmolality. And some of these are actually possible to measure on board nowadays. And just to give you an idea, this is more like a class slide, how they actually behave in time, all of them. Also to mean that we can actually measure them at the beginning of the stress and the end of the stress, while in the middle of the stress phase. And that's one of the problems that we don't have. We don't know when the fish started to be stressed, and for many species we don't have basal levels. That's also something that we need to work on, have baseline levels for some of these species. Okay, so starting with some of these case studies. Here you see the set nets. This is the typical catch. As you can see, these are relatively small boats, so not much space to have gear, welfare-related gear, stunning mechanisms, sometimes not even ice. And you can see here how the fish are actually stored while they are collected. So you could see some of the fish still pretty much alive in those trays. These are trips that are made off the coast of the port cities of Portugal. So they go out, they stretch the net, then they recover the net, and then we measure what's going on. So here we measured 4 different species. And what I want you to look at here is that depending on the species, the vitality at arrival on board, it's quite different. So the sparids have quite a high vitality, but the mulids have a very low vitality when they arrive on board. So again, a species-specific question. The other thing that is important to look at this is that if they arrive well on board, they will suffer for longer on board because they will die later. So sometimes we think, no, this is a good thing. Well, it was a good thing until they are hauled on board. Then it becomes a bad thing. With these indicators, we were also able to create models where we can relate specific conditions in the water or in the atmosphere, for instance, air temperature and depth and water temperatures, and relate that to the loss of vitality on board. To relate that to scale loss, I'm not going too much into the numbers. I just want to show you what it can be done. Here I'm showing also you some details about 2 species, the common 2-banded sea bream and the red sea bream. And in the end, you see that cortisol on arrival of those that are at a high vitality is always lower than cortisol on arrival of those that are in the low vitality state when they arrive. So we probably have been struggling for more, but not always the indicators show exactly the same thing. And this is important for the fact that we are using several indicators. But in general, activity or demise correlates well with osmolality, for instance. which is always a good indicator, and it shows that overall there's a systemic imbalance in these fish while they are demising. Okay, so here I'm going to show you briefly the second example. You see Vignesh there. This is long line, so long lines can be up to 4 kilometers in these small fisheries. The boats are relatively small, 6 meters. Again, not much room to have anything there, but they are about 50 to 60% of the fleet going for the long line. So these really small boats. This is also because our coast is relatively shallow, and our what's the word in English? Our continental shelf is quite shallow and quite long, so that's why you have this kind of fisheries there. For the long lines, I'm not even going into the graphs. I'm just saying that glucose depletion correlates with loss of vitality. Lactate increases significantly while on board because of probably to hypoxia and anaerobic respiration. But those species that maintain high vitality, they have accumulation of cortisol, so they're still increasing their stress response while on board. And again, osmolality is correlated with these changes, especially in the first periods out of water. For the purse seine, so here we use a much larger boat. In fact, this is the largest boat in our southern coast, going for purse seines. And I just want to show you how the method is compared to the purse seiners here in Northern Europe. And because the catches are not big enough, so this vessel, it has a pump as well. They could pump the fish. In fact, we selected this vessel because we wanted to compare brailing and pumping. But it was very difficult to convince them to use the pump because they say, well, we only use the pump if there's a big catch, and we never have big catches. So in the end, we ended up testing the brailing and the conditions of storage on this vessel. So we tried to, I don't know if you noticed the scarce use of ice they are putting on these boxes. And so what we tried to see was how the fish would respond to be stored under ice, on ice slurry, and try to change some of these practices. See, you have Anna here taking blood from a sardine, but the target species that we used for this study were mostly Scombers collies and Sardina pilchau, so the chub mackerel and the sardine. And here, what we did was to look at vitality for the two species on the fish collected on the first braille and the fish collected on the last braille. So it's 20 minutes in time between the first and the last braille in general. So in these conditions, all while in the tank as in the normal conditions. And vitality is still very high between the first and the last braille. It goes down very fast once the fish are put in the tanks. But if we use ice slurry, it's interesting because vitality goes even faster, okay? So it drops much faster. Okay, this is not a sign of stunning. This is not a sign of better welfare, but it's a sign that the fish will not be jumping around and probably getting even more injuries. So in all conditions, but especially in ice slurry, the vitality decreases much faster. And I'm going to show you just the example for cortisol, although crowding increases the levels of all physiologic indicators. Cortisol in the first braille, cortisol in the last braille, cortisol of fish in tank, and then cortisol of fish in the ice conditions, either from the first or the last braille, and then either after 10 and 20 minutes in these conditions, and here in ice slurry, which is much better. All of these fish from here, they were collected in the first braille. Okay? So what it shows is that, in fact, if you store them in ice slurry, you stop the accumulation of cortisol in this species. So whether this actually, again, changes the awareness of the fish, or it's just a metabolic consequence of being in a very cold condition and all the stereogenesis, etc., is much slower, we don't really know yet. So just to summarize this, combining our approaches, the biological functioning approach, effective and natural, which we have not done, but looking at the usefulness and limitations, we have, if we use the first one, we have quantifiable indicators, so something that we can actually measure, but requires some sampling. So it's a mix between having good data and having fast data, and no baseline values means that we cannot compare specifically to each species how it was in the beginning and how much the values that we are measuring, they actually mean for the species. In this case, so the effective safe makes welfare more relatable to the fishers, which is also important. We want them to understand how the fish is behaving and what it means to the fish and how it can relate to the fish. But of course, they're hard to measure in these conditions. And of course, here we would probably try to look at fish that escape or that are released to look if they can maintain their survival rates. But this is something that we have not done. So just to wrap up this part, having all these criteria or these indicators means that we have triangulation, we get information from all sides, we can cross-validate the different indicators, and we can have scalability in the sense that we can go from what is inside the fish to the behavior and what it would mean for the fish if it was, for instance, released. And with that, we try to improve welfare standards. Outcomes of the project, so the outcomes are public in these 5 reports. So these 5 reports, they describe the fishery, they describe the project approach to the fisheries, and then they have key points on capture, handling, and sending and slaughter, how to improve these fisheries. With this, we built recommendations and guidelines, and then we turned this information to Friends of the Sea to provide welfare standards, and Mario will talk about that tomorrow as well. And again, you can see this in the posters outside. One outcome that I want to highlight here is the Fair Fish database. And the Fair Fish database scores the different species and the different fishing methods into the current welfare based on scientific and grey literature, on the what would be the potential welfare if some changes are done, and what would be the worst case solution for each species. And with that, just to give you an example, so what Jenny has been doing with other collaborators in the project is to see what are the welfare hazards of these fisheries, then score these fisheries, the current, the better situation, and the potential, or and the worst case situation in terms of scoring. So of course, the longer the blue bar, the better the welfare. And with that, you can also see some mitigation measures in the database. And this is an ongoing process. So many fishes are already being prepared, and many more will be done. And now, moving a little bit from this project, I want to present you what the welfare for catch, advancing animal welfare in fisheries, which is a more focused project, again on small scale fisheries, and here also having a worldwide partner, which is the Sustainable Fisheries Partnership. Will try to bring our discoveries to their examples that are globally recognized, and they also have a very wide database. What we want to do and what we will do, so again, measuring welfare, but also now tackling more the underwater situation. And the other important part that is CCMAR job is also to take practical and smart gear to the fishermen. Again, as you've seen the example from below and the examples for these small-scale fisheries, it's very difficult to put things on board, and we have to make it practical for the fishermen to want to introduce them in their daily practice. And with that, I mean, I think we all agree that that a lot of these changes will go through consumer empowerment, educational strategies, and improving practices, addressing consumer demand, etc. And I thank you very much for your attention. Not so bad.
Okay, we have time for one question. No, nothing for Pedro?
Everyone wants coffee. I
yeah,
want coffee.
there's maybe that. So, if you give everybody your name.
Hi, Alex Chailloux from Cine in France. I was wondering if keeping the fish alive in water tanks on small-scale fisheries, it sounds doable easily for them, as they have pumps with the engine running. is a solution which is envisaged sometime, because auctions could sell the fish live. That's made in some places. It doesn't appear as a solution which is sort of imagined by things you have presented.
Well, that would be, for some fishermen, that would be a very, what's the word, a very attractable solution, because we have a saying in Portugal that comes from older, which is sardinha viva, and that means live sardine, because people would, the fishermen would get to the beach with the sardines jumping on the boat, and that's when the consumers wanted to buy because it was fresh. Of course, the problem is if you keep them alive, you're just increasing their suffering in a way, because they will be dead at some point. And that would be possible in bigger boats, because the catch, as you can see, I mean, just imagine that tray that you have there, what's the density of fish that you have to have in a box or in a tank alive to keep the normal density. So that would be very difficult to do in terms of welfare, but also in terms of practicability. The boats are really small, and everything else that you put a pump, anything else just takes more energy. So more batteries, something else more, it would be more complicated. Of course, in time, this will probably expand into having bigger boats with more technology, slightly bigger boats. One thing that I was looking from the crab fisheries, that you have small boats in Scotland, but if you look at these small boats, they are very, well, I wouldn't say high tech, but they are very well prepared with all these gear, technology, etc. And if you look at some of the boats I showed, it's like they're not really that well prepared, and people don't care that much. So it's a big change. So that change that I first showed in the first slides in very small letters. In fact, they should be in very big letters for these kind of fisheries.
Right, thank you very much, Pedro. It was an excellent talk, as ever. So, and what you've been doing in that project is advancing catch welfare forward. Huge steps. So thank you very much.
Thank you.